Okay, as I explained you before, in this forest, which is the Shonai coastal forest, we have two sides. One is the pure side, and the other one, the video that you saw before, this one is the mixed side. In the mixed side, we have two species. One is the black locust and the black pine, as you saw already. And this one is the pure side. So here we only have black pine trees, which is the one you can see here. As before, they have an inclination because they are a windbreak forest, so they are pushed in the direction from as the wind, So the wind is coming from the ocean, and then here is they are pushed into that direction. So here we have 5 trees that we sample 3 times in a year, in summer, early autumn, and late autumn, because the nitrogen and phosphorus at the same time is quite dynamic and changes during the year. So they move from roots to leaves, and then from leaves to roots to stem, and then they fall to the ground as litter. Here I will show you some litter that we have. For example, the leaves don't fall one by one, but they fall in cohorts, as you see here. So there are leaves of different ages, actually. But the ones that fall the most are the leaves that are 3 to 4 years old. Here you have older leaves. You can see them because of the color. The color is, they have loose that green that they have when they just fall this year. And the nitrogen and phosphorus that is here is released to the ground when this organic material is decomposed. And that nitrogen, in this case in particular, goes into the ground as ammonium first, and then is nitrified into nitrate. Here, this particular tree uses the nitrogen that is recycled, let's say, in a closed cycle. So all the nitrogen that is being uptaken is produced by the same tree, and of course is also falling, there's also atmospheric deposition, but mostly is the nitrogen that is being recycled by the same tree. And we know that because of the nitrogen isotope that we measure in these trees. We took samples of the soil, we took samples of the litter, we took samples of the roots, the stem, and the needles, and the leaves in the case of black locust. So we have followed not only the nitrogen in the soil tree medium, but we have also taken tree ring samples to understand how has been the development of nitrogen acquisition of these trees for the last 20 to 30 years. This study has been done mainly by Kazushi-kun. Kazushi-kun is an undergraduate student in my laboratory. He's graduating this year, and the results that he has obtained for this study are really, really good. The measurements are very clear. They show us very clearly how the nitrogen is moving in a pure forest, and how are interacting in a forest that is mixed, and mainly the influence of black locust trees, which is a leguminous species, on the black pine trees. Today I asked Kazushi-kun to come with us to the forest. As you can see, it's already January here, so there is no understory vegetation, only branches. So when we come here in summer, it's quite difficult to walk inside because there's lots of vegetation and understory vegetation. And here, after this forest was established, there was also, of course, an increase in biodiversity. So there are many insects here and small animals, but one of the insect that is probably most representative here that I always see is a big spider that has the colors of a bee. And we also always have to be very careful because it's quite big, and there's always between this side. So Kazushi-kun, here we collected, as I mentioned before, all the samples that we needed for the study, but one that was I don't want to say the most important, but one that show us how the tree has been uptaking nitrogen in the last decades is the core sample that we take from the stem. So for that, we use this instrument that is called increment borer. It's an instrument made by a Swedish company, Haglof, and it's very simple, but this is an instrument that only that company makes, and even though many countries have tried to imitate it, nobody has been successful. So we still buy this instrument, and now Kazushi-kun is going to show you how he takes the sample and how that sample looks like later. Okay, Kazushi-kun.
はい。
So this forest is, we consider it as a flat area. Of course, it's almost at the sea level, but as you can see here, it's not as flat as we actually think it is. There is still micro relief. There is a micro topography that also has to be taken into consideration when we compare one tree with the other. And also, as you will see in the presentation that I will do about Kazushi-kun's study, the soil is very sandy. So the capacity of the soil to retain nitrogen or phosphorus or water to that matter is very low. So even if you put lots of let's say fertilizer in the soil, the capacity of the soil to retain it is very low. Now Kazushi-kun is going to take the sample. We use this spoon, which is used to extract the sample from the tree. So once we take the sample, we put some glue, as you can see here. This is glue to close the tree. Now Kazushi-kun is going to extract the sample. And this is the tree, the sample. So this is a black pine tree. I don't know if, well, the cork is very thin, so it's almost disappears, but you still can see there. We bring this sample to the laboratory, and we use a sandpaper to clean it, and then the trees are very clear, and when they're very clear we can scan them and use a special software to count the tree rings, and then based on that image, based on that reading, we then cut the rings individually and measure the nitrogen nitrogen isotope and content in every ring. Now here, if carry this one approach, you can see here the center of the tree. So from here to here, you can count how old is this tree. It's not very clear maybe at the moment, but I will say that this tree is probably 25 to 28 years old. So this is a young tree. It's not as old as the ones, the next one. We just use it for an example at the moment, just to show you how we take the samples. All right, well, this is all for today. Please enjoy the presentation. Kazushi-kun has done wonderful work. So I will, after the presentation, if you have any question, Kazushi-kun or we can answer any of your questions. So I leave you with Kazushi to say goodbye.
See you!